Hi everybody, we are going on to video V41. This video deals with some other mechanisms of inheritance, the ones that Mendel didn't discover. So let's go right ahead and jump into this. Obviously our slide here is supposed to be semi-humorous. Remember that Mendel used peas because they were fast growing. Redwood trees aren't. Okay, that's funny. So Mendelian genetics. Understand, or let's recall what it means when we say something has been inherited in a Mendelian fashion. To say that it's inherited in a Mendelian fashion means that there are two alleles per trait, right? We saw that when we looked at the alleles. We had big T, or big S, little s. You had two alleles per trait, and one was dominant and one was recessive. You also had just one gene per trait. That is, there were not more than one gene contributing to a particular trait, like height or flower color. There was simply one gene involved. There was no environmental influence. We didn't see any effect when Mendel was describing his work of, well, this color only appeared when it was this temperature or this moisture. And there's no sex difference in expression. By that we mean there is no male or no female difference in how a trait's inherited or expressed. So please understand, Mendel did not discover all hereditary mechanisms. He did not come up with all of heredity. Inheritance patterns are often more complex than those that Mendel discovered. Mendel was quite fortunate he picked peas to look at because they have some traits that are inherited in a very straightforward two alleles, one dominant, one recessive, one gene per trait, no environmental influence, and no sex difference in expression system. So other mechanisms, though, of inheritance are more complicated and a little more involved. Or you can think of this as these things would have driven Mendel crazy. So, well, I should show up here. Let me show up. How you doing? So these are some traits that are inherited in ways that are not going to be how Mendel would have understood them to be. So this collectively, we call these traits non-Mendelian to demonstrate that they are inherited in a way that Mendel would not have been able to predict or understand based on what he saw. So let's just go through these. One type is called incomplete dominance. So the heterozygote in this case has its own phenotype. Recall before when we had the other Mendelian traits, if we had big T, or big T, little t, they had the same phenotype, the same appearance. Here we're saying the heterozygote has its own phenotype. In other words, there's no dominance here. So this would be one type, this would be another type, and then this, the little t, would yet be a third type. So rather than two phenotypes, would be three. And often the phenotype is somewhere in between the phenotypes of the parental varieties, so it looks a little bit like blending inheritance. So what we're saying here is that a dominant allele does not subdue or take over or dominate. Dominant makes it sound like it's actually dominating and beating it up. The alleles don't interact. Think of it like this. If we're talking about a trait, let me go ahead and show you the example. And let me erase this, of snapdragons. You have red snapdragons and white snapdragons. These are just kinds of flowers. So, of course, all the gametes that the red ones can make, and they're showing you as R's and W's, don't be confused by that. If you want to think of it as, C big R and C little r, you could do that too. Don't be confused by the W. But here we're saying all the gametes are going to fuse in the F1 to form, I'll go ahead and use this system again, C big R, C little r. So they're all heterozygotes and they're all pink. And this looks kind of like blending, doesn't it? Because you would have red and white. If you mix red and white paint, you'd get pink. So you're thinking, aha, this is showing you something a little bit different. And the reason you get that is if you have one version of red, you make some red pigment. If you have white, you make white pigment. So you mix some red and some white, and it does show up as pink. However, how can you prove it's not blending inheritance? If you were to cross this with itself, so if you were to cross C big R, C little r with C big R, that's a sad R, C little r, I'm going to have, these are my big R's, they're red. I should get half of my offspring being pink, and I do because these are the heterozygotes, and then I get one that's white. So this actually is blending inheritance. Again, an example of a feature that does not work like Mendel would have predicted. So again, it's an example, let me move, it's an example of non-Mendelian inheritance because you don't get the ratios Mendel would have predicted. What would Mendel have predicted? A 3-to-1 and in the F2 generation, and we sure don't see that here, so it's got to be non-Mendelian. So that's one violation, if you will, of Mendel's traits, because he always had one dominant, one recessive allele in his traits. Mendel also only saw 2 alleles, big R, big T, little t. There are some genes where there's more than 2 versions of them, and one example is in blood typing. There are 3 alleles in blood typing, and they write them a little differently here. They'll have IA, IB, and just I. If you want to think of it as IA, IB, and IO, you can think of it that way. And notice then there are 3 alleles. And you're like, that can't be 3 alleles because I only have 2 parents. There can only be 2 alleles. That's true. You may only have the A and the B, or a B and an O, or 2 B's, or 2 O's, or 2 A's. But the population has multiple alleles in it. And clearly, if you have that being the case, when you try to look at ratios from that, it's sure not going to be what Mendel would have predicted. For example, in this case, we have 4 different phenotypes, and notice also we have an example of codominance, because if you have A and B together, and I'm right here, then you have its own phenotype. If you have A, or A and O, you have A blood type A. We have blood type A. If you have B and B, or B and an O, that's blood type B. If you have 2 O's together, you have O. But again, I have 6 genotypes, and just so you can see that, make sure, okay, I have 1, 2, 3, 4, 5, 6 genotypes, but 4 phenotypes. Once again, Mendel would not have understood this. He would not have understood why he was getting this many, because he had a system where you only had 2 alleles per gene, and here you have more than 2. You have 3 in this case. So, once again, same message. You don't get predicted ratios. This is an example of a different kind of inheritance than Mendel understood. Okay, here we go. Keep going. Environmental influence. Mendel's never saw a case, come over here, Mendel's never saw a case where he had to worry about the environment changing the phenotype. But there are some genes that change their expression based upon the temperature. This is the Arctic fox. The Arctic fox temperature changes, the fur changes depending upon the temperature. So the environment can influence the phenotype. If you have somebody who, let's say they're halfway starved, they're very poor and they don't get a lot to eat, that's going to change their phenotype because it's going to change how their genes express. So they may not be as tall, or they may be more malnourished because of that. The environment can affect the phenotype. So in this case, Mendel wouldn't have understood how he could cross a white and a white fox and get this color depending upon the time of the year. That was, again, was not something that he studied. So, let me give you this formula down here to show. This is just how scientists talk, so sorry. But we say VP equals VG plus VE. And that sounds really stupid. But VP means the variation in the phenotype is equal to a genetic influence and an environmental influence. So for some traits, like you see here, The environmental influence is really dramatic, and that's going to change how the phenotype looks. So VP equals VG plus VE, not BG, because they were a group back in the 70s. Yeah, sorry. Okay. Here's another example, hydrangea. Got some of these in my yard. The pH actually influences their expression. If I believe this is correct, if they're pink, it means the soil is sort of acidic. If it's blue, it means they're sort of basic. So the same genotype is going to change its expression depending upon the environment. So once again, Mendel wouldn't have expected this because he didn't see any of this in his peas. Okay. Yet another example. This one and one more. This one's called polygenic inheritance. Okay. Now listen carefully. Multiple genes can affect a trait. We've been talking before about one gene affects a trait, like one gene affects height, or one gene affects a flower color. Here we're saying you can have multiple genes which all contribute to a phenotype. Now, when you have quantitative characters, are those that vary in a population along the continuum. In English, that just means you can have some traits that are like this, where you have very few individuals on one end, very few individuals on the other, and a lot of people in the middle. Think about human height as our example. There are very few people who are very, very, very short, and there's very few people who are very, very, very tall. Most people are sort of average. Whenever you see a trait that if you graph it looks kind of like this, I'll try to draw it better down here, like a bell curve. That's not much better. If you see a curve like this, that usually indicates there's more than one gene influencing the phenotype. And there are some examples of this that we'll mention. Okay. Again, if you look at height, this is a famous picture taken back in the early 1900s at a school. And if you look at the height, they lined up based on the number of people who were that height. They found this kind of relationship that strongly suggests that there's more than one gene involved. You see that same thing down here. Another example is human intelligence. There's very few people that are up here by IQ that are above 145. There's very few people by IQ that are on this end that are below 55. Most people are kind of in this average range, but the fact that you have someone here and someone there and someone there and someone there suggests more than one trait's involved. In other words, we're not all either brilliant or stupid. You have variation, and that's because intelligence seems to be influenced by multiple genes. Okay, and we're almost there. Well, forgot I had another example. Skin color is another example. We believe there could be 5, 6, 7 different genes that contribute to skin color. This is showing you, let's pretend for a second, just for giggles, that there's 3 genes involved because they're showing you A, B, and C. If you get this version, then you get no melanin, so you're going to be way down here. If you get this version, you're going to get a lot of melanin, a lot of skin pigmentation, and you'll be here. But very few people are really here or here. Most people are going to be in the middle, some range. This is actually the easiest combination to produce from this couple, so that's why it's the most common. Okay, and again, I have the same message each time, which is, notice again, if you're Gregor Mendel, you can't handle this because you would not expect to have a range. In this case, you have 7 different phenotypes. He expected to see 2. So that's why it's an example of non-Mendelian inheritance. Okay, our last example deals with what's called sex linkage. There are some traits that are inherited differently based upon our gender. So here in our example, we have a female and a male. Okay, it's easier for a male to end up with a sex-linked trait because he only inherits his X chromosome from his mom. to be a male, he has to inherit the Y chromosome. So let me draw one here for you. Okay, so this is a guy. Okay, here is a trait that we're talking about. So you can see half of the males that are produced here are going to get the blue version, and therefore they're going to be unaffected. Half are going to get this version, and they're going to be affected. But notice why they're affected. The X chromosome does, I'm sorry, the Y chromosome does not have a version of that trait. So whatever X chromosome he gets from his mom, if he happens to get this one, he has the trait. If he happens to get this one, he does not. Okay? For females, it's actually harder to get it because you have to have, your dad has to have the trait, and your mom has to either have it or be a carrier. So one of the main characteristics of sex linkage is these traits are more common in males than they are in females because there's more ways for males to get it, or it's easier for males to get it than for females. And again, the reason why is most likely due to the fact that the X chromosome is this nice, big, robust chromosome that has quite a few genes on it. The Y chromosome, by comparison, has fewer on it. It does have a gene on it that's important for determining maleness, so that's why it comes as an important trait for males to inherit, to have the gene for maleness. But they don't have, if you're talking about a trait that's here, obviously they don't have that on their chromosome, so whatever version they get from mom is the version they have to express. So some sex-linked traits are associated with disorders, and we'll talk about some in a second. And just to recap, males are at much greater risk for inheriting these because they only get one X, the X from their mom. So if that X happens to have the allele for the disorder, then they're going to suffer from that disorder. Some examples of this are male pattern baldness, red-green color blindness, myopia, night blindness, and hemophilia. For example, if you have a male with red-green color blindness, he looks at this chart, he cannot distinguish. You might be able to see very easily the 15 in there. The males who have that condition can't distinguish that. Hemophilia, obviously, it's much more serious. Myopia, we won't get into all the visual disorders like night blindness or myopia, but there's quite a few where males cannot see correctly because they're inheriting versions of the alleles from their mothers, and since they just get that from their mother and just the Y chromosome from their dad, then they have to have the condition. Punnett squares can be used to predict this, and we'll do some of these in class so you can understand. But notice on this, and again, it's a little confusing, there's the Y chromosome, and the Y chromosome doesn't have an allele on it for the reasons we just said. Y chromosomes don't have alleles for these. So here's mama up here. She has X big B, X big B. Here's the father. He has an X little b and a Y. So this case, because you have two X's, that's going to be a female that could be produced, and she's going to be a carrier. This is going to be over here a female who's going to be a carrier. Here is going to be a male who is not going to have the condition if it's recessive. Here's going to be another male who's not going to have the condition if it's recessive. But let me show you over here just for a second. Okay, if I have this case, if I have a female who's X big B, X little b, and let's say the dad is X big B, Y. So let's do the Punnett square. That's not a very pretty square. You tried trying to draw with a mouse. Okay, so let's look at the possible offspring. Again, this is the female. Okay, this is the female up here, and this is the male. This is the dad. Okay, what are the gametes, or what are the offspring they're going to produce? Okay, a female, X big B, X big B, that's a female without the trait. Okay, here's going to be a female, X big B, X little b, who's going to be a carrier for the trait but not have it. Here's going to be a male, X big B and Y. Okay, so that's going to be a male who's okay, but here's going to be a male produced, an X little b and a Y, so he's going to have the condition because he inherited that version from his mom, and since the Y didn't have anything on there, that's what he's got. Okay, so just to recap, we looked at multiple examples of non-Mendelian inheritance, polygenic inheritance, where you have more than 2 genes involved, multiple alleles, where you have multiple versions of alleles involved, like in blood typing, incomplete dominance, sex linkage, and environmental influence. So please, one more time, understand, Mendel is famous because he figured out what he did with Mendelian inheritance. But there's lots of examples of inheritance that don't work quite like Mendel discovered. Okay, we'll wrap up here. We'll do an assignment. Thank you very much.